So we're starting a new unit today on intracellular compartments and transport, and that may sound kind of blah, but I actually love this topic and I think it's really cool. And when I came back around to teach this class, I thought, man, this is cool. And I vaguely remembered hearing about it in my cell biology class, but what this is getting at is we've talked about how proteins get made. We talk about how proteins generally act. And then we went and talked about some membrane proteins, how they generally act, what they do. Then we went through how does the cell get energy to make those proteins operate. This is talking about how do cells get the proteins to where they need to go in the cell. Because every protein that a cell makes has a particular location. Sometimes that location is going to be in the cytoplasm, but if it's supposed to be working in the cytoplasm, it needs to be in that location to do its job. Or it may need to be in a particular membrane or inside of a particular compartment. So the way that cells get proteins and other molecules to the correct place is the topic of this particular unit. So this is the transportation patterns of the cell. And when we talk about proteins going to this organelle and that organelle, you're going to see that it's a lot of hassle and it's a lot of work and it takes a lot of energy for the cell to get the proteins to the correct location. So I want to start with the question, why bother having organelles if they're that difficult to maintain and expensive to keep operating? More organized. It is more organized.
The cell can't function with all of them, without all of them.
The cell can't function without all of them. Some cells do. Microbes do. So yes, more organized. Cells that need organelles need them to operate. What's the benefit of organelles? Complexity and compartmentalization. So if you want to be doing several different functions at once, you need to be more organized. If you want to have the possibility of different metabolic conditions in your cell, you can enclose those specialized functions in an organelle. So keep these activities in here separate from the activities that are going on out here. And when you think about the concept of pools, so what I'm writing on the side over here, the pools, now that you have heard a lot about metabolite pools, having separate metabolite pools is important. So if you have a molecule in the cytosol, and you also have that molecule inside a lysosome, it could be acted on by different enzymes in those compartments. It could be in different conditions. And all of that together does allow more complexity. That's part of the reason why we see that eukaryotes that have a variety of organelles can do more complicated tasks than prokaryotes. That's part of the reason. So it is worth it to have organelles, even though it's expensive energetically to maintain them. Probably at the beginning of the next lecture, I'm going to show our favorite video again with the molecular animation. And one thing I love about that is it showed how crowded the cell is. It's even more crowded than that, but they did a pretty good job of showing how much stuff is inside the cell. And so from this point, when I talk about cytoplasm, I want you to know the specific definition of that is the cytoplasm includes the cytosol, liquid gel structure, plus all the stuff that floats in it. So everything between the nuclear envelope and the plasma membrane, that would be the cytoplasm, and the liquid component, which is actually a little more viscous than just plain water, that's cytosol. And the trick for a cell is, how do I navigate getting molecules to the correct location through all of this stuff? started to say chaos. It's actually not chaos, it's organized stuff. How do I get it to the correct location? This is for you to just look at later. I'm going to talk about compartments in general. So we're going to talk about the nuclear compartment. We're going to talk about the endoplasmic reticulum specifically. And then I talk about later organelles, the further out organelles, like the more distant planets or something like that. So generally we're going to be moving proteins along a path from nucleus, cytosol, ER, then to these other locations. And remember, every protein that's heading to these locations, it needs to be going to a specific place to do its job. So we're going to start with the nucleus because really we're looking at central dogma here. So we've got DNA being transcribed to RNA, RNA translated to proteins. And we talked earlier about what it takes for DNA to be transcribed. We had RNA processing there, then the RNA comes out to the cytoplasm and gets translated by ribosomes. So we need to say a little bit about what's going on in the nucleus at the start. The nucleus, most of the time, if the cell is not dividing, and you look at a nucleus through the microscope, it looks fairly round and fairly diffuse. Like you don't see condensed chromosomes, you're just seeing fairly relaxed chromatin in the nucleus. And there is also a membrane around it that you can't see with our microscopes. But that can be called the nuclear membrane, or it can be called the nuclear envelope. Different books will call it different ways. I actually use both terms consistently. So nuclear envelope, nuclear membrane. And it's thought that we know that eukaryotic cells originally derived from prokaryotic cells, which don't keep their DNA in a nucleus, but they do have it attached to the membrane at some point. It took a while for scientists to figure that out because usually the DNA is so much bigger than this, but it is attached to the membrane of the prokaryotic cell. So it's thought that at some point in the incredibly distant past, the plasma membrane invaginated, that's an excellent word, invaginated, come in and folded around the DNA and then formed the nuclear membrane. I should write that on back there. So it is enclosed by a membrane, but this membrane that is around the nucleus is more permeable than your average membrane. So what we're going to look at next is what it is and is not permeable to. This slide I think I want to skip over. It's just showing that the organelles also have membranes around them that probably was from engulfment instead of invagination. Okay. So actually we come back to the nucleus in a minute. So we have the DNA in the nucleus. We're doing transcription of RNA in the nucleus. I'm going to pull this up for a second. So we're doing transcription in the nucleus. We're sending messenger RNAs out here after they've been processed. Hang on, I'm going to draw that on the other side. All right, we're having messenger RNAs sent out to the cytoplasm, and they're translated into proteins. Every protein is translated, at least starting to be translated in the cytosol. So that's sort of the central location where ribosomes are found and can do the translating. And then those proteins, they need to get to where their location is. There are a lot of possibilities of where they could go. Some of them need to go back into the nucleus. Like, what's a protein that needs to be in the nucleus? DNA synthase? DNA polymerase? Good guess. Yes, so stuff that makes more DNA. What else? Some cofactors?
The things that help DNA.
Transcription factors.
Transcription.
Yeah, transcription factors. Histones would need to be back in there. Pieces of RNA polymerase need to get back in there. Several things. What kind of protein would need to go into the chloroplast? Rubisco. All the parts of rubisco. What kind of proteins would need to get back into the mitochondria? Get into the mitochondria? Complex blank. Right? Cytochrome C. Krebs cycle enzymes. A lot of stuff. Certain ones to the peroxisome, certain ones to the interior of the ER. So one thing that will be coming on our next exam is I want you to think about that. Like, don't just think about these processes in isolation, but think about, for a particular protein, cytochrome C, its whole life history, so to speak. So we've got a gene for it. We made messenger RNA for it. It was translated. Then it had to get where it was going. Then it does a job. Then it might eventually get degraded by a protease. But all of that is required for it to do its job. And the reason I focus on that is sometimes you can make a protein like, this is just fine. You made it perfectly, but it didn't get to the correct location. And that can cause a disease. The gene that made golden number 4 right there may be fine. There's nothing wrong with it. It's translocation or its transport that is messed up. So connect this. This stuff gets very abstract. Connect it with individual proteins. Every individual protein that we need needs to go through that pathway to get to its correct location. Here's how the proteins know where to go. They don't know. They're not sentient. I'm doing so many words today. What was that other word? I'm going to forget them. I don't write them up here. Here's how proteins know where to go. They have signal sequences. And the signal sequence is expressed in amino acids. There will be a set of amino acids somewhere in the protein primary sequence that serve as this signal sequence, and they're like a barcode on a package. Is our best example from human world, from people world. So these are translated along with the protein. And so when you see something like this, this is the N-terminus. This is the N-terminus of the protein. So this part would be translated first. So the first thing that comes out is this sequence. And for that particular sequence, and where it's showing green, it has to be, the colors mean something, but it has to be generally like this. That's a signal that this protein needs to go into the inside, the lumen of the endoplasmic reticulum. When you see ones that have carboxyl group on the end, that's the C-terminus. So this particular signal sequence that says stay in the lumen, don't even come out again. Just stay in there, okay? Just stay in there. That's translated last. So a protein that's going to go in would have this at the N-terminus. It may or may not have this at the C-terminus, but if it does, it's signaled to stay in there. When you see ones that don't have N or C-terminus listed, those are interior signal sequences. Like those are just in the middle of the protein somewhere. Not on the N or the C-terminus. So these are like FedEx shipping barcodes. It's like, oh, this is supposed to go to Memphis first. And then it will go to Bristol or whatever. Those are the signal sequences. I don't need you to memorize what the signal sequences are, but just the concept of them. They can be N-terminal, they can be C-terminal, they can be interior. But what they have is the information to tell the protein, this is where you're supposed to go. This information, the other thing you need to know about it, is that each one of these tags have a 3D shape. It may not be a fold, like the really short ones. This is too short to be a fold. It's just 3 amino acids, but it would have a 3D shape that is identifiable or bindable. Something like this, it literally does fold into a detectable shape. So the shape is coming from the sequence, whether or not it folds. The real recognition comes from shape. Finally, if you end up in research, you can use this information to make a protein go to a new place. You could engineer a protein that doesn't usually go into the peroxisome. You could make it go in there just by putting this tag in it. So that can be useful. So I want to talk about first proteins that go to the nucleus. The protein in question has this particular tag, which indicates that it is supposed to travel to the nucleus to do its job in the nucleus. And when proteins are going back into the nucleus, they must cross through the nuclear envelope. So the nuclear envelope, it's actually, I don't want to say it's double membraned, but it's kind of, there's a lumen to it. And then you have the interior of the nucleus, and out here you would have the cytoplasm. The nuclear envelope or the nuclear membrane has pretty large size pores in it. This is not the kind of membrane that's going to keep out sodium, let's say. This is like laundry basket, or it's got some large channels going through it, nuclear pores. So the nuclear pores, when you look closer at them, they have a very bizarre structure. I don't know, I just kind of love this. It looks like a mess until you understand how it works, and then it all makes sense. It's a great structure allowing the function relationship. So here's the nuclear envelope. That's the membrane part. And it's got these pores, these holes, and the pores are lined with particular proteins, shown here in yellow. And they have a lot of unstructured secondary structure in them. So these probably have beta sheet and so on. These may have some alpha helix. This mess in here, this is unstructured. It's not doing alpha helix, it's not doing beta sheet. It's unstructured amino acid chains sort of filling in this pore. And then on the inside of the nuclear pore, there are these rod-shaped extended proteins that are forming this basket, nuclear basket, formed out of these nuclear fibrils. I don't need you to know the individual names of the proteins. not in the yellow. There's some later I need you to know. But generally, there are fibrous proteins sticking out in the cytoplasm. There are fibrous proteins sticking in. And this whole thing, to me, looks like one of those Frisbee golf basket. Like you could throw something in there and then it stays in. An insect could fly through it, but an eagle would get trapped. Why would that? Silly. It's kind of like a Frisbee golf basket. One other thing I want to point out is that there are these proteins that are holding up the nuclear envelope. That's called the nuclear lamina. One of you has a talk that's related to that. I think it's Caitlin, maybe, one of your cell papers. Nuclear lamina we're going to look at in the next section. And if you do excellent electron microscopy, this is what nuclear pores look like top down. They're pretty because they're symmetrical. They look kind of like stars. And from side on, this is technically astounding that anybody could get this picture, but there's the envelope, there's the basket. The fibrils are too small to be resolved in this. So that's a strange looking structure. The reason that the structure is this way is that it allows very small molecules to pass in and out. But it can be selective for protein passage. It's selective for protein passage. And we're going to look at how it's selective for protein passage. That's just a closer up view. I'm going to skip through that. All right, here's a picture that has some new information on it. And in this picture, I do want you to know the cherry donut and the blue thing. Okay, I'm going to talk about what those are. So here we have what we're calling a prospective nuclear protein. This has a function that's supposed to be happening in the nucleus, and there is its signal sequence, like a cherry on top. It would be on the outside. It's at the N-terminus. It's the same sequence that we saw in this chart earlier. So, where did it go? This. It's not N-terminal. Sorry, it's not N-terminal. It's this sequence. And that sequence is going to be forming a shape, a detectable shape, on the exterior of the protein called the nuclear localization signal, or the NLS. And it's permanently bound into this protein that's supposed to go into the nucleus. This NLS is detected or bound, or you can think of it as almost like a ligand, I guess, for a protein called nuclear transport receptor, which is the blue thing here. And the nuclear transport receptor, if it has cargo, if it's bound to an NLS and the protein that that's attached to, it has better affinity for the pore complex. So it's kind of like, if it's not bound to cargo, it floats around in the cytoplasm until it encounters its binding partner, and then they stick together. So the same forces, hydrophobic, hydrostatic, van der Waals, that make those stick together. When those are bound together, that allows this to interact with the nuclear pore complex. And it can go through the unstructured region. So there's more detail to that, but for this class, these two bound together may pass through. It's kind of like, I don't know, you can't go through this subway turnstile unless your parent is with you, something like that. There they go inside. And inside, something needs to get these apart. So they have good binding affinity for each other. Something needs to get them apart. There's an enzyme called RAN GTPase that separates these. So many of you misspelled separate on your column chromatography that I just did it too. It's SEPARATES. I kept circling that. You're leading me astray. Separate. That spins a GTP to do so. So it takes energy to rip these two apart. When this is interior, without a binding partner, it heads back out. It goes back. I don't have a picture of that, but yes it is, it's on here. When this is alone in the interior of the nucleus, it has binding affinity for the other side of this, and it will go back out, pick up cargo. So maybe this is like an airport shuttle or something. I go into the airport, I drop off my passenger, I come back out, I look for more. There's a separate enzyme in here that makes those two come apart. Something to think about. RAN GTPase itself had to come in this way. It would have a cherry on top. It would have come in here with this protein, which is kind of interesting. I guess another one had to come along and get them apart from each other. But that's how proteins get into the nucleus. They go through this strange nuclear pore complex with the help of a nuclear transport receptor. Sometimes, this is not always the board figure, but it's a candidate that it could be a board figure for an exam.
So if there's a protein that's like destined for the nucleus and it has to leave and then come back every time.
What do you mean if it has to leave and come back
Like
every
it,
time?
well, like it's made in the nucleus, right?
They're made in the cytoplasm, right? Yeah.
Okay.
The RNA for any of these would be made in the nucleus. Go out here to be translated on a ribosome. The ones that have to come back would come back this way after they're translated. That'll be more obvious in a minute because I kind of skipped over that for these. Mitochondrial import, I'm not going to go into as much detail about. Remember, we did talk about the outer mitochondrial membrane can let small things that are less than 5 kD through that membrane. The inner membrane is more normal for permeability. But proteins that have to get in here have to cross both membranes, which is a pain. There aren't many proteins out here. I mean, what would be there? What's usually in here that we learned about? A lot of you labeled it on your test. That would be here. Oh man, that's so good. We got protons out here. We got a proton gradient, but there's not too much else that has to be in here. Let's get rid of that. Lots of enzymes need to be in the matrix. Enzymes that need to get, or other proteins that need to get into the matrix, they have a signal sequence that directs the ribosome to bring them over here, and they get connected to this receptor protein. And that receptor protein, this is bizarrely cool, moves this protein laterally until it lines up with channels in both membranes. And then it threads through. So one general concept from this is if proteins must cross membranes, much easier energetically to thread through the backbone. So cells have developed these systems for getting proteins to cross membranes by threading them through a small hole before they fold. Instead of letting this fold and trying to push that big thing through both membranes, I mean, then you would have to have these huge holes. This system of threading a backbone through a small hole and then letting it fold, that's how cells tend to move proteins across membranes. And so in mitochondria, you've got to have two holes lined up together to get the protein to go through both membranes, get into the matrix, and then fold up into its final shape. This process I don't make a big focus on. I do want to make a big focus on how this works in the endoplasmic reticulum. This is a really good stopping point. So let's have a break here before we get into the next details.